Good morning, student. In previous lecture of chapter respiration and energy transfer, in that chapter we discuss the anaerobic respiration. In that anaerobic respiration, we discussed the definition of anaerobic respiration. Then anaerobic respiration take place in glycolysis. We also study the process of glycolysis. It is an incomplete type of respiration. We discuss detail about the anaerobic respiration. Now, in this lecture of the chapter respiration and energy transfer, we discuss the next type of respiration, that is the aerobic respiration. Firstly, we study the definition of aerobic respiration. The respiration in which molecular O2 is involved is called aerobic respiration. Or we can also define the respiration which takes place in presence of molecular O2 is called as aerobic respiration. The aerobic respiration completed in a following step, that is a glycolysis, acetylation and Krebs cycle. We discuss one by one. First, the glycolysis. Glycolysis is the same as in anaerobic respiration. We know very well that in glycolysis, glucose, which is a 6-carbon containing compound, converted into two molecules of pyruvate, which is a 3-carbon containing compound. This is same a common process, or the glycolysis is common process to both aerobic as well as anaerobic respiration. Now, the second step in aerobic respiration is acetylation, or it is also called as the conversion of pyruvate into acetyl coenzyme A, or it is also called as a link reaction. Now we study the second step in aerobic respiration, that is acetylation. Firstly, the acetylation process takes place in the outer chamber of mitochondria, or we know that the process of respiration takes place in mitochondria. The first step, that is the glycolysis, takes place in the cytoplasm of the cell, while the acetylation, which is the second step of aerobic respiration, and that acetylation is take place in the outer chamber of mitochondria, and then the last step, that is a Krebs cycle, take place in the matrix of mitochondria. Now, the acetylation means simply conversion of two molecule of pyruvate into acetyl coenzyme A. Here, the pyruvate undergoes simultaneously dehydrogenation and decarboxylation due to loss of CO2 and H2. The pyruvate get converted into acetyl coenzyme A. The lost H2 is accepted by NAD to form NADH2. Due to loss of H2 and CO2, the pyruvate get converted into acetyl coenzyme A, which is a 2-carbon containing compound, and this process is called as acetylation or link reaction, because it link between glycolysis and Krebs cycle. Now, next to that, the last step, that is the Krebs cycle. Now, in case of Krebs cycle, the acetyl coenzyme A is completely oxidized and CO2 is released in stepwise manner. It's called Krebs cycle. Also, the first stable compound in Krebs cycle is citric acid, which is 6-carbon containing compound. Chemically, it is a tricarboxylic acid, and hence Krebs cycle is also called citric acid cycle or tricarboxylic acid, or shortly TCA. Now, here we studied the Krebs cycle. The acetyl coenzyme A now enter in the matrix of mitochondria. In matrix of mitochondria, various enzymes as well as coenzyme is present. Firstly, the acetyl coenzyme A enter in the matrix of mitochondria accepted by oxaloacetate in presence of water to form the citrate, which is the first stable compound and 5 carbon containing compound. The citrate by dehydration converted into cis-aconitate. Again, that cis-aconitate by hydration converted into isocitrate. The isocitrate by dehydrogenation means again here loss of H2 from that isocitrate. Due to that loss of H2, the isocitrate get converted into oxalosuccinate, and that loss H2 is accepted by NAD, forms the NADH2. Now, the oxalosuccinate by decarboxylation means loss of removal of CO2. Due to removal of CO2 from oxalosuccinate, that oxalosuccinate converted into alpha ketoglutarate, which is a only one 5 carbon containing compound in the Krebs cycle. Now, this alpha ketoglutarate simultaneously by decarboxylation and dehydration means simultaneous loss of H2 and loss of CO2, the alpha ketoglutarate converted into succinyl coenzyme A. Here, succinyl coenzyme A by hydration means water molecule is added, and due to that some amount of energy is released at the time of succinyl coenzyme A react with the water. At the time some energy is released, and that energy is immediately accepted by ADP and forms a energy rich molecule, that is the ATP. Means here one ATP molecule is also forms. Now, due to hydration, succinyl coenzyme A converted into succinate. Again, succinate by dehydration converted into fumarate. But here the lost H2 is accepted by coenzyme FAD to forms FADH2. Now the fumarate again react with water and get converted into malate, while the malate by dehydrogenation means again from malate H2 is lost, and that H2 is accepted by NAD, forms NADH2. Due to loss of H2 from malate, the malate get converted into oxaloacetate. Here the Krebs cycle is completed. Here in this lecture we discuss the acetylation as well as the Krebs cycle. Simply, acetylation means conversion of two molecules of pyruvate into two molecules of acetyl coenzyme A, which is a 2 carbon containing compound, and the acetyl coenzyme A enter in the matrix of mitochondria, where oxaloacetate react with that acetyl coenzyme A to form the citrate, and citrate by dehydration converted into cis-aconitate. Again, that cis-aconitate by hydration get converted into isocitrate. Isocitrate by isocitrate by dehydrogenation means loss of H2 converted into oxalosuccinate. Oxalosuccinate as a decarboxylation converted into alpha ketoglutarate. Alpha ketoglutarate by simultaneously dehydrogenation as well decarboxylation converted into succinyl coenzyme A. Succinyl coenzyme A react with water to form succinate. Now succinate by dehydrogenation converted into fumarate. fumarate react with water to form malate, and lastly the malate by dehydrogenation again converted into oxaloacetate. Here the Kelvin cycle which is the last step with aerobic respiration.